Well, as people are rolling in, why don't I go ahead and get started and introduce the time. So welcome everybody to the Friday Research Seminar. We're glad you could be able to join us for the final college research presentation for 2021. Our presentation today is by Zoom. So just a few housekeeping things. We've muted all of your microphones, and we'll be using the Q&A button below in terms of questions that you may have for the presentation. I will be moderating the Q&A. And by the way, if those of you who don't know me, I'm Emily Ho, faculty in the nutrition program in the college. So I will be, again, moderating the questions and repeating the questions to our guest speaker today. With that, it's with great enthusiasm that I'd like to introduce our speaker for the final seminar of the term. I personally think we've saved the best for last. But today joining us via Zoom is Dr. Penny Gordon-Larsen. Dr. Gordon-Larsen is the Associate Dean for Research at the University of Carolina Gillings School of Global Public Health. She started that position in 2018, but she has been at UNC for many years. She started as a postdoc, one of the Dannon Institute postdoctoral fellows in 1998, and then joined the faculty in 2002. Just this year, she was honored for her work in the field of nutrition with the Carla Chamblee Distinguished Professorship. Dr. Gordon-Larsen is truly one of the nation's leading nutrition science researchers. Her research is focused on obesity. Much of her research focuses on issues related to ethnicity, disparities, and the development of obesity over the life cycle. She really appreciates that obesity is a complex, multifactorial disease, and she's really been a thought leader in multidisciplinary and, more importantly, transdisciplinary approaches to tackle big problems in our health, like obesity, by integrating both molecular and genetic approaches to environmental and also societal level factors that influence health. She has a very long list of awards, but in particular, she has served as past president of the Obesity Society. She's received a Scientific Achievement Award from the Society as well. She serves on the NIH NADDK Advisory Council and co-chairs the Strategic Plan for Dissemination and Implementation Research. So we're really pleased that she's able to spend some time with us. Today, she's going to talk about her work in obesity and the multifactorial approaches that integrate biology, behavior, and the environment to understand obesity and its cardiometabolic consequences. So welcome.
Thank you, Emily. Thank you for the very kind introduction. And I'm really happy to be here to talk with your faculty and students. And as Emily said, I'm going to be talking about the biosocial influences on obesity and cardiometabolic diseases. And as you're well aware, obesity has become a really major global problem. And you can see here from the 90s to the 2010s that there's been just a tremendous increase across the globe in obesity. And the obesity continues to be an issue largely because of the way we think about it and manage it. And the prevailing view has really been that obesity is the result of simple thermodynamics and just a simple issue of energy balance. And researchers have by and large investigated obesity in very separate and distinct lines of research. So either it's basic or it's clinical or it's analytical or it's population. And they generally approach obesity asking very specific questions about specific and separate aspects of obesity. And it's this lack of transdisciplinarity that has held us back. And the traditional approach ignores the situation of energy balance here at the bottom of this figure within the larger socioeconomic framework. And when we think about the, from the top level, the societal factors that house the obesity, the community factors that influence obesity, and the interpersonal, and how all of these interact with the behaviors that are key for obesity. So showing here the diet, physical activity, sleep, sedentary time. and how those then interact with individual susceptibility, like genetics, hormones, microbiome, and so forth, that lead to energy balance. And what's critical here is really that interaction across levels and the interlinkage and flow in both directions, from the individual susceptibilities up to the societal factors and back through. And what I'll attempt to do today is really talk through how we can think about this kind of nested framework. And so I'll start at the top level first, and then go deep. So my early work, this was a paper that we published back in 2006, looked just like, in the past, at very single factors. And so this is looking at built environment factors, in this case, looking at the education of the population and how that related to the allocation of physical activity resources in neighborhoods. And here this showed that areas with higher educated populations were more likely to have physical activity facilities. But this work really is fairly limited in its focus just on this one kind of factor, and really ignores the complexity that underlies that distribution of resources across geographic and social space. And so one of the biggest issues that faces neighborhood research is the fact that people choose their environments. And so the problem is that for any given person, if you value exercise and you want to live in an area that supports your exercise, it becomes very difficult to disentangle your health consciousness and preferences from the causal impact of the neighborhood supports on your behaviors. So here's two examples of papers, one by Pasquale Rumo on the left and the other Katie Meyer on the right. Pasquale was a former student who's now at NYU, and Katie Meyer is a faculty member at UNC. And in these two papers, we used complex modeling strategies to dig into this issue of unmeasured confounding and try to disentangle the influence of individual choice on the neighborhood impact on behavior, and trying to understand which direction causality flowed. And one of the issues with this kind of work, even though it's methodologically complex, is that there still remains a pretty big amount of individual variation in the effect of neighborhoods on individuals. And so, for example, you can think of somebody who is going to be physically active no matter where they live. And then you can think of somebody else who, really chooses an environment because of its amenities, and then goes to that environment and winds up not taking advantage of the amenities and not engaging in physical activity. So all kinds of individual level variability play into those factors and further add to that complexity. So we looked then to try to understand some of the susceptibility factors that play a role in how the environments influence individuals. And so in these two papers, and I'll present a little bit more deeper on these, we looked at urbanization as the environmental pressure, and how the massive environmental change in China influenced individual molecular level factors, such as the microbiome and the metabolome. And so this paper on the left, which is really about the microbiome, was by a former student, Catherine Wingley, who's now at CDC, and Yuqing Wang on the right, who just graduated from UNC and is now doing a postdoc at Harvard. So when we think about how this works, and we think about human biology, we have the ability now to measure tons of molecular markers to help understand these complicated pathways. And so to do that, we can start with each of the components and steps along the way. So looking at the effects of environments on those molecular markers, and then whether the relationship between the environments and diet or obesity, for example, are leaving molecular marks. So we operationalize those measures through various omics measures, such as genomics, proteomics, metabolomics. And this gives us a sense for what is happening under the skin. And as you know, these kinds of assays and profiling result in a tremendous amount of data. And one of the major challenges really is understanding how to manage that huge data volume, integrate it, and interpret it. And what we can do with that flow, particularly if we have multimodal and multi-omic data sets, is look, on the range all the way down on the left for the genomic side, which can tell us what could happen. The transcriptome can tell us what appears to be happening. The proteome can tell us what makes it happen. And then the metabolome can tell us what has happened and what is happening. And early studies used omics, but had serious limitations. So some of the work that really prompted the work that I'm about to show you in China comes from these urban-rural contrasts with the microbiome. And so what individuals did, the scientists did, was take samples from very rural areas and compared them to samples from very urban areas. And what they did was pick samples from across the globe. So they took hunter-gatherers in Africa, for example, fishing populations in the Philippines, for example, industrialized populations in Europe, and compared them and made inferences about the effects of those environmental pressures on microbiota. And one of the really big challenges there is that they were not just comparing urban-rural, but they were comparing then different geography, different climate, different cultural traditions across the globe that lead to very different diet patterns, very different kinds of exposures. And so that limits what you can do with some of these complicated molecular markers. So what we decided to do is take advantage of the massive change in China. And China has had this very dramatic urbanization that's unfolded more rapidly than any other place on our planet. And this is just, for example, showing you just one example in Shandong Province in the same village over time. In a very short period of time, they completely retooled an entire community. And they do this across China to look from a very traditional kind of community to a much more modern community. And these changes just happen, in a matter of a few years. And so that change allows us to take advantage of longitudinal sampling to look at what happens when communities have that kind of massive transformation. And we know that the urban environment in China in particular is incredibly densely populated. And with urbanization, there's an influx of very modern, highly processed foods, a lot of reduced active transit, a lot of exposure to pollution, and just very different kinds of new and modern lifestyle kinds of pressures. And so for our China project, we were very interested in how urbanization influences diet, obesity, and cardiometabolic risk factors. And we capitalized, as I said, on that massive change in lifestyle and environments that we see in China. And so these green areas on this map show the provinces that we've been studying and following for the past year as part of the China Health and Nutrition Survey. And these circles are red, in red here, are communities where we have specifically sampled within the same communities very rural and very urban environments in very close proximity. And so for this very saturated sample, we're looking at 4 provinces: Hunan, Guizhou, Guangxi, and Henan. And within each of those 4, we have a rural sample and an urban sample. And then the squares are megacities in the that are part of the China Nutritional Transition Cohort Study. And for that, we have fecal samples and 16S RNA sampling of over 2,000 individuals. And so those megacities are Beijing, Chongqing, and Shanghai. And then the rest of the green are the other provinces in the study. For our saturated sample, we have 16S RNA. We also have whole genome, metagenome sampling in a smaller cohort. And then we layered on plasma metabolome so that we could get a view of the metabolites that would change with urbanization. And in this paper led by Shan Sun, she's a student that I work with from UNC Charlotte, she found very large variation in our gut microbiota across provinces. And so going down in these different colors on the left are the different provinces in our study. And the circles that you can see, that all of the small shading is just the general variability in the full sample. And the circles that you see are the clustering of the metabolites by each of these provinces. And so what you can see here is that for these circles, for each of the provinces where we see that distinct clustering, that there's very little overlap across each of these provinces. And that actually differs a lot from what you see. We did a comparison where we looked at the Human Microbiome Project in the United States, where they're sampling across all counties of, well, not all, every single county, but there's county level sampling across all states in the United States. And we see basically very little clustering, that for the most part in the United States, that we have very similar microbiota, whether we live in Oregon or North Carolina or New York or North Dakota. Whereas in China, we see this very distinct clustering. And in the right, this is a view of the variance that's explained, and you can see that province is responsible for most of the variation that we observe in the microbiota, and that demography and demographic factors are also really, really key. And you can see just as you go down the levels that diet and physiology are actually kind of not that important in explaining variation compared to province. So when we delve into our urban-rural sample, you can see again that distinct clustering, and these colors correspond to province. Red is Henan, blue is Hunan, green is Guangxi, and orange is Guizhou. But the shading that you see, like the red, you see two shades of red, the different color shading is the urban versus rural, and that's the same for each of these colors. And so you can see that there continues to be variation, even though there's differences from province to province. There are also differences within province by urban and rural. And you can also see clear clustering on when we look here at this view using OPLS-DA methods to cluster our samples. And you can see that there's clear clustering on the vertical plane for urban versus rural in the red and green, and in the province level with Henan on the left. Hunan on the left and Guizhou on the right. And then when we delve into this a little bit more to look at the relationship between the microbiota and its association with diet, and with some of the other risk factors that I'll show after this, that we can see that here, looking at sodium in the top left, potassium below, and then the sodium to potassium ratio, that we can see that there are associations that differ for each of those nutrients. And that these ones that I've highlighted are pathogenic bacteria in yellow, short chain fatty acid producers in blue, and then bacteria that have been linked to cardiovascular disease risk factors in green. So clearly some of these really important factors are popping up and suggest that there might be some pathways here that we might want to take a look at. Similarly, we found metabolites that are associated with, in the top panel, sodium, and the bottom panel, potassium, and then on the right, the potassium to sodium ratio, and that some of the metabolites that light up for their associations operate in the obesity and hypertension related pathways. So again, giving us a sense for how some of these mechanisms might work that for the effects of diet on cardiovascular disease risk factors. So ultimately, using these untargeted metabolomics and the microbiome data, we found that both microbiota and metabolites differed by blood pressure. And so all of this put together really gives us a lot that we can look at as we delve deeper. So again, each of these investigations was really focused on a sliver or a very small part of the puzzle. And now, as we're developing this more evolved perspective where we recognize obesity as a disease and as a complex multifactorial disease that really requires an integrated and translational approach. And this is an editorial that we wrote just a short while ago for Circulation in response to their the American Heart Association's policy where they were calling obesity a behavior. And so I think we're getting more and more information about how complicated obesity is and how much more there is that we can do. And it's really become clear that we need to look at obesity from the translational perspective. And so this is a figure from NCATS, the National Center for Advancing Translational Sciences, which is leading the way here. But this is a really great figure, I think, for showing how we need to turn observations in the laboratory, clinic, and community into interventions. And how we need synergies across these different levels. So we really need to be doing more work where we talk, crosstalk with basic scientists, clinical scientists, and public health researchers. And so I'll show you a couple of examples of how I think we can go about doing that. So the premise for the work that I'm going to talk about today is that you can have two individuals who maintain identical diets, but one develops the disease obesity and the other does not. And one reason may have to do with the complex biochemical processing of food that may differ on the basis of genetics or on the microbiome. And those differences can translate perhaps to genetic inefficiencies that block the processing and lead to more energy harvest from the diet. So, you know, explaining why two individuals might have the same diet but differ in their body weight. Or really a multitude of very different factors that can differentially impact each person. So we go back to this energy balance equation and look at it from a more complex perspective. You can see that there's a lot of individual variability in the susceptibility factors or the individual predispositions ranging from the genetic, epigenetic, all the way through early life events. And that they interact in complex ways with environment and lifestyle factors that influence behaviors, which then have an influence on that energy balance. And a variety of evidence suggests that the response to treatment varies widely across individuals. So while mean weight loss can be favorable for a weight intervention, there's a lot of variation and there's a lot of differential response to treatment. Some people gain weight on a weight loss treatment, some people stay weight stable, and some do wind up getting the desired weight loss. And what we want to do is use the individual predispositions and the environment and lifestyle factors to find the right treatment for the right person at the right time. And genetics can play a role in an individual's response to diets and in their likelihood of developing obesity. And so what we really want to know is the genetic contribution to the diet responses. And preclinical animal studies can help us to decipher genetic links. But one of the biggest challenges for that kind of work is how does the mouse really translate to the human? And typically, a lot of the mouse studies use these very genetically identical mouse models. So the clone, specific mouse model for a specific disease, for example. And one of the problems with that approach is that humans are genetically diverse and have varying responses to diet. And so we want to get a better representation of that diversity in preclinical studies. And so the approach that we're using in our group is to use a set of genetically diverse mice. And so these are diversity outbred mice. And this is a model of genetic heterogeneity. And basically they take these 8 genetically identical strains of mice that vary wildly on their various properties like body weight. And then they randomly breed them and crossbreed them. Then they result in mice that are genetically diverse and unique. And so then you can do experiments with these mice to look at how they respond to different kinds of treatments. And then you can pick up on some of that variability that predicts some of that differential response. So what we're doing here, and this is work that I'm doing in collaboration with Steve Hursting and John French, and we're doing diversity outbred mouse study where we induce obesity. So it's a diet-induced obesity model. We have 12 weeks of very high fat diet, which 60% of calories is fat. And then we have a switch in those same mice to caloric restriction for 8 weeks. And that's switching to a 6% fat model. And these are the results. These are individual curves for male and female mice who have been part of the study. And you can see the break from the high fat diet to the caloric restriction here. And each of these lines corresponds to a different mouse. And so you can see that for some of the mice, they have a tremendous gain in weight, and then they drop off at the caloric restriction. You see other mice that have a tremendous gain in weight, but then they don't lose that weight on the caloric restriction. And then you see others down at the bottom who are impervious to the overfeeding or underfeeding. They're just weight stable regardless. And we see similar variation in HOMA-IR. Here, when we look at the baseline, they are variable because they're, that diverse stock there. But when you look at the experimental condition, you can see a tremendous amount of variability in that response. And so from those responses and using the genetically diverse mice, we can see which of the precise locations on the genome may be associated with the outliers and get a sense for the general trends in weight gain and weight loss. And so as an example, when we analyzed the differences in insulin resistance in the mice, we were able to pinpoint a position here on chromosome 12 that was potentially responsible for some of the responses that we saw, the insulin resistance responses to that experimental condition. And so what we can do then with that signal is really take that signal into the human studies. And so that is taking this preclinical study to in the mouse model to the human study. And when we do that, going from mouse to human is not a new idea. People have been doing it, but I'll show you how we're doing it and how we hope that we're making inroads in really informing this kind of translation, where we can, take the mice and do the kinds of experiments that you just can't do with humans. You can't really overfeed and then energy restrict large populations of humans to understand some of these impacts. So the plot on the right is the locus zoom plot. And so we're looking for the evidence for association of glucose and insulin resistance in human populations. And the red rectangles here on this figure are showing the 3 genes in the region that are homologous between mice and humans. So there is some translation there to humans. And the GWAS results are from the MAGIC study, which is the meta-analysis of glucose and insulin-related traits consortium. And that's a study of over 15 150,000 study participants, humans. And so we're going to be doing this comparison between the mouse and the human population. And the candidate genes that we find that are homologous on chromosome 12, they, for the mice, correspond to chromosome 14 in humans. And directly underneath that insulin resistance signal, there's sign 2, which is a GWAS signal for sphingomyelin, for lipids, and for insulin growth factor 1 levels. For PPP2R5E, that is a protein phosphatase regulatory unit, and that is a GWAS signal for height and blood pressure. And then there's KCNH5, which is related to dopamine and dopamine receptors, and it's a GWAS signal that we see for BMI and subcutaneous adipose. And this plot shows the homologous genes and the gene ontology based predicted protein interactions. And so you can see for the mouse to the human that the genes show a very similar predicted interaction contributing to insulin resistance and high fat in diet-induced obesity. And so we've got these signals, we've got these pathways, and now what we're doing is moving to the mechanistic and/or knockout studies to validate. And in humans, what we'll do next is the fine mapping of the glucose signals. We'll look at additional related phenotypes, and we'll bring in more omics so that we can add more information about what is happening with the gene regulation. For mice, we're considering additional phenotypes there as well as omics. But for mice, we can do things like look at tissues. So we can look at blood, urine, liver, pancreas, visceral, subcutaneous adipose to hopefully get us a better understanding of some of these signals. So now that we have the signals from the mice, what we do in humans where we can't do these kinds of experiments is look at weight and weight change. And one of the things that we need to do to be able to do this work is to think about heterogeneity and how we capture heterogeneity to be able to tease apart some of these subgroups that we find differential susceptibility with. And so how do we do that? How do we capture that heterogeneity in weight? So some of the questions that we think about is, is it fat distribution that matters? Is it the time, the duration of obesity that you've had? Is it the amount of extra body weight that you have over time? So we're capitalizing on longitudinal data from the Atherosclerosis Risk in Communities, or the ARIC study. And here we have data from over 15,000 individuals over 25 years. And so they were 45 to 64 at baseline, and they're followed over 25 years. And we have measured anthropometry and lots of really dense phenotyping on these individuals. And so this is work that I'm doing with Annie Green Howard, who's a biostatistician who I work very closely with, Carrie North and Christy Avery, who are epidemiologists. And so we have a lot of this anthropometry. And so we take that anthropometry, and what we do is pull it together into a single measure that captures duration, intensity, and fat distribution, which we operationalize as classes. And so this is a latent class analysis to get us to this single measure. And we came up with 4 groups, starting with moderate baseline, small gain, and then a low baseline, large gain. And interestingly, those 2 groups start off around the same baseline weight, but have very divergent paths. High baseline, small gain, and then a high baseline, large loss. And we found variation in the sociodemographics that are associated with those classes. So a higher proportion of women in the 2 classes that end up highest. We have a higher proportion of African Americans in the 2 groups that started off highest, but then diverged. And a higher proportion of highly educated individuals in that stable low group. And then a higher proportion of smokers in the really divergent groups, the group that increased over time and the group that decreased over time. And then we ask, how are those different classes associated with health outcomes? And here we're very interested in the cardiovascular disease related outcomes. And so to do that, we looked within fasting blood, we looked at a GWAS, we looked at plasma metabolites, and then a range of the typical CVD risk profile markers, the dyslipidemia, glycemia, blood pressure, et cetera. And then because these folks are followed through the period at which they experience cardiovascular disease events, we have events, myocardial infarction and fatal coronary heart disease that we also look at. And this is a complicated figure, so I'm going to kind of go slow through it, but basically the classes correspond to the figure in this upper right panel. So you've got class 1, the red curve, class 2, 3, 4 here, and then I'm going to show you a baseline, which is the darker bar value here at the top for diabetes, then hypertension, inflammation, and triglycerides. And so I'm going to look at these comparisons. But so what we find here is that, there's heterogeneity across the different classes, the different subtypes of obesity that we see in relation to the cardiovascular disease risk profiles. So class 1 had the largest jump in diabetes. Class 2 had the largest jump in hypertension, so more of a hypertension profile. Class 3 had a high on just about everything. And class 4 had drops in inflammatory and lipid markers. And so we asked how those subtypes were then associated with the future events, myocardial infarction and fatal coronary heart disease. And we found that there were relatively more events in the high group, this high group, as well as in the rapid increase group. And then the lowest percent that we saw was in that stable low group. And then this group that tanked had the highest proportion of fatal events and major events. But what we wondered was, what is the deal with this group that tanks? Is it that they were sicker to start with? And we found that there were no differences in baseline kidney function, nor were there differences in baseline cancer rates. So we also looked at whether genetics could help us tease apart the difference in the subtypes. And so we found a hit on chromosome 11 here. And when we go to zoom in here, we see that the signal that we pick up is correlated with a signal that's reported by Lee Kaplan's group in this paper by Hatum. And this paper was a paper where they looked at the genetics in relation to successful bariatric surgery outcomes. And we found that this signal that we find in the population-based approach that is associated with differential outcomes here was a signal that is associated with success in bariatric surgery outcomes. So again, giving us some insights. When we look at the metabolome differences by group, we look at the patterning of association between metabolites and hypertension. And you're not meant to see the chemical names here. I'm going to zoom in on the top 20 in the next slide, but here this is really just to show you the qualitative differences in patterning that you see across these 4 classes, with the darker colors representing the more negative association with hypertension. The darker red is the more positive associations with hypertension. And you can just see that the patterning of these associations is incredibly different across classes. If this were randomly distributed, we'd see things looking more alike across those classes, but instead you see really clear differences in that patterning, again suggesting that there are subtypes of obesity that really have differential kinds of underlying molecular markers and cardiovascular disease risk outcomes. When we take a closer look at those top 20 metabolite hypertension associations, you see not just the darker colors in class 3 and 4, but you can also notice the very differential effects that we see in direction across all of these metabolites and across the classes. So something's going on that's really different here. And these top 20 that we see in their association give us some insight as to some potential pathways and some potential signals that we might want to follow up on. So, so I hope that that has given you a sense for, how we can approach looking at energy balance from a translational perspective, and how we can think about how the individual susceptibility factors relate to the behaviors that are then housed in these higher level kinds of contextual influences. And all this work is really starting to point at subtypes, and we see subtypes in our outcomes where we can see heterogeneity in the outcome that can let us divide up susceptible groups for understanding who might be most at risk for disease. We can also see heterogeneity in the types of environmental exposures, and in our case, looking at those urbanization factors and how those differential environments may impact individuals. And then in the susceptibility and pathways, by looking at the molecular markers, the genetic markers, the microbiome, to really get us to a point to integrate and understand some of these complicated relationships. And I'm grateful for all of the funding over the years, the many institutional and national funding and support that's made a difference to me, and more importantly, an impact on understanding of obesity. And so what I'll say to this is that also, like many senior researchers, the road to all of this funding, which is really impressive and looks amazing, was paved with lots of grant rejections, lots of non-discussed grants, lots of papers that were rejected from journals. So I've gotten really lucky over the past 20 years with my success in some areas, but lots of unsuccessful ventures that you haven't had the opportunity to see. But just a message to say that where you don't get things discussed, or you don't get the award, that you just have to keep persisting and trying to push through and hopefully get that success. So I will end there and answer any questions that the group might have, and happy to hear any of your thoughts. Thank you.
Thank you, Penny. That was amazing. Lots to think about, and certainly a very tangled web in terms of things to think about. So just a brief reminder, if folks have questions, to use the chat button that's in the middle, or sorry, the Q&A button that's in the middle of your screen. And there are a few questions that have already popped up, but I thought maybe I would start with a broader question, especially thinking of some of the graduate students and early career trainees that may be in the audience. Can you talk a little bit about doing transdisciplinary work and approaches to really better position taking a transdisciplinary approach to these research questions?
Yeah, so I think the biggest thing with doing the transdisciplinary work is really having a sense of humility and not being afraid to admit that you don't know something and to ask questions of your collaborators. And when you find the right collaborators who are also themselves, have their own dose of humility and their ability to really, answer those questions and ask the same questions, that you can ask the really, kind of naive and questions that lead to greater understanding across the fields. And sometimes being able to start from scratch, really, with your collaborators who speak often different languages, to be able to get on the same page. So, what I've done in all of these cases with the large transdisciplinary teams is really spend a lot of time in the beginning for us to get shared language, and then for us to ask each other questions. And a lot of our conversations really do wind up kind of starting off with, this may be a stupid question, but, And I think that eventually we get to a place where we can understand the perspectives that each of us has and really help us get there better. Sharing methods is also really important. When we did that work with the identification of subtypes based on the latent class analysis, our mouse modelers had never heard of that, even though it's a pretty common technique for population scientists. And so they were very excited about it. And we actually wound up paralleling the latent class analysis work with the mouse studies at all. I didn't show that today, but we identified subtypes in the mouse data as well. And you know, for them, because it was something they had never heard of, was really exciting for them, and you know, leads them to do a whole different kind of line of research. So I think you know, really being able to pull people together to spend the time talking, and to just have that dose of humility is really key there.
Speaking of being able to talk, I guess, with different groups, there's a question in the chat in terms of, do you have any tips for nutrition professionals, for example, and what they can do to have a more culturally recognized way of helping the population or their patients with obesity, especially for underrepresented communities like the BIPOC community?
Yeah, so I didn't talk about my work in health equity at all, but that's a really huge issue. I think one of the biggest things that we can do for obesity is really to forward this idea of obesity as a multifactorial disease. Because what happens when you think about BIPOC communities is that, you know, there's a whole lot of intersectionality. And when you are obese and Black, for example, or you know, obese and rural, or you know, whatever it is, and you wind up suffering not just the bias of whatever your skin color is, or whatever kind of societal biases that are out there, but you suffer the double bias against obese individuals. And people with obesity have a tremendous amount of bias, both in the societal judgment and bias, but also in all kinds of barriers to healthcare and treatment, very poor treatment, particularly if you are a Black woman who is obese. You get less pain medication, you get less, you know, a lot of pejorative comments from doctors and so forth in general. And I think what we can do by talking about the genetics and the molecular signals and the differential response is to shed light on the fact that, you know, energy, it's not energy balance in and of itself. And that you can't just tell somebody to eat less and move more and expect them to be able to have the response that you would like to see, because of that complexity. So some obese people consume very little calories, and it's not the fact that they are consuming a lot of calories, it's the fact that there's something in their physiological or biological system that is causing you know, very challenging situation for them to be able to lose weight, keep weight off, manage a healthy weight, et cetera. And so that's one of the biggest things I think that we can do. The other thing that we can do, I think, is really to fight against that bias and to fight against the barriers to access to treatment. A lot of communities just simply don't have access to high quality obesity care. And we know that we need that. And you can, as you know, you can really increase understanding of the fact that you should have a board certified diplomat in obesity medicine who is providing, you know, a comprehensive obesity medical care. When you have that kind of care, you'll get some, you know, a physician who will partner with a lot of different kinds of providers to talk through medications that have weight gain side effects, to remove those. They'll address stress. They'll address your barriers, your environmental situation, your structure. So it's not just about diet and exercise, it's about sleep, mental health, a whole range of other factors. And so making sure that people understand that that is a part of high quality care, and to really argue for more services out there to support that care is really important. Similarly, to argue for and advocate for policies to increase insurance coverage for weight, anything related to weight management access across the population. So I think those are the big things that I think we can do. And I think it's important for us to think about the heterogeneity that is in relation to subpopulations of, and particularly disadvantaged populations in our society, so that we can get a better understanding of the differential impacts and barriers faced by, all populations, so that we aren't ignoring any slices of the population, and that we're making sure that our vulnerable populations receive access, but that they aren't further, biased and put in situations where they just, have further prejudice that they're experiencing.
Good points. Following up on your answer, and also related to a couple of questions in the chat, is circling back to, you mentioned stress and mental health, and those as environmental factors that may affect obesity, diabetes, but then also the reverse. How does obesity affect mental health and stress as well? Can you comment a little bit about that?
Yeah, so I do not study mental health outcomes myself. So I do very little research in that area. What I can say is that mental health is a clear and important part of the package, and that the more people that can bring the mental health and obesity world together, the better. We know that mental health, as I mentioned, a lot of the medications, particularly medications that are related to or that are prescribed for mental health issues, do have weight gain as a side effect. And so, at a very bare minimum, getting the substitution for those medications is really important. But we also know that obesity has effects on on depression, on social stress, on all kinds of stress. And so, and occupation, there's a lot of occupational discrimination that happens if you're obese. And so, it's a cycle of further reducing the economic wealth of obese individuals while adding to their adverse mental health outcomes. And so there's just a lot going on there, and I think really important to tease apart. And I just encourage as many people as possible, if they're interested in that area, to pursue this area, because we definitely need more people doing that combined work.
Again, a very tangled web of
right.
information and inputs and outputs. There is also a more specific question about some of the first part of your talk, focusing on the geographical environment and microbiome. And this question wants a little bit more in terms of how do these specific and certain environments affect the microbiome, even in a healthy context?
Yeah, so we know that there's a lot we don't know about how the microbiome relates to the health outcomes. One of the things that we see, that was shocking to me in our China sample, is that when we look in our China sample, I mean, even if we look in the rural environments, I mean, typically we think our rural environments and more traditional lifestyles are healthier environments. And so certainly from a dietary perspective, they're, usually associated with more healthful diets. But even in those communities, we saw indicators of the kinds of microbiota that are associated with adverse outcomes. So there were a lot of viral genomes that we picked up in our whole genome metagenome work that are suggestive of antibiotic resistance. For example, the genomes that are those bacteria signals that we found present. And so it suggests that as we see, these environments that have effects on the microbiome, that as we see the development of urbanization in our case, even at the very low end, that those changes in the environments really wind up having a big impact on what happens at a molecular and a microbial level. And one of the things that, that I mentioned is that in the United States, across our country, that we have very similar microbiome profiles across our country with very little regional variation. And what we think is happening, even in that kind of healthier environments in China, is that with urbanization, that we get this homogenization of bacteria. And that lack of variation, I mean, we know that consuming a variable diet results in a variable microbiome. And we know that variability helps us in terms of a variety of health outcomes. So variety is best. And we see this constraining and homogenization of the foods we eat and the bacteria we house, which then gives us very little to work with in response to different kinds of insults. So, it's very interesting that even when not directly associated with disease or with, adverse environments, that we do see this processing that that seems to suggest, kind of an evolution that's not that helpful for us based on the kinds of westernized lifestyles that we're leading. I don't know if that really answered the question, but that's how I think of it.
No, that's great. One final question. As we have seen the scale of and the technologies in terms of the number of things that we can measure in a given population has really exploded, especially over the last 5-10 years. Where are we in terms of our ability to integrate that data in a meaningful way?
Yeah, we're in the infancy. So we're just learning now, how to do that well. There are some really wonderful methodologists who are developing integrative omic techniques to be able to do that. There's a lot of work in machine learning and kind of the decision science area as well, that's looking at some of this integration that really what we hope we would be able to do is take the sampling from individuals, the molecular sampling and the genetic sampling, and to be able to very rapidly analyze how these different kinds of data intersect to result in kind of the profiles that might benefit best from specific kinds of therapies so that we can guide those therapies from a more precision angle. But there's a lot we need to do. That's probably, the kind of next frontier for the data. We've got the technology that has been kind of put out there before the analysis and integration has really been fully developed. So there is definitely a lot to do there.
Thank you. Well, I really want to thank you for your time, your insights. It's a really fantastic and an insightful presentation. I'd like everyone to join me in thanking you again for a fantastic presentation. Wish you a great end of the term, everybody, and good luck with your finals and your grading, and hope everyone has a happy holidays.
Thank you. Thank you all for coming, and thank you for hosting me.
Absolutely. Thank you again.